And thank you, the CGDG group, for the invitation. I know this is an unusual topic for the Broadley Institute, where people work with a bunch of things, but mostly animals. So thank you for coming and joining us. So the idea is to go through the genetic data analysis process we've advanced for the past decade or so, kind of showing where we are now and what's going to happen next. Again, my name is Guilherme da Silva Pereira, but people call me Gui for short. And we are going to go through a brief introduction about what being a polyploid means. And I split the topics into where we are now and what's next, basically showing our latest advances with examples for potato and sweet potato, and talking about what we are planning to do next, and mentioning the things we've developed here so far during my sabbatical. So I've been working with polyploid since my PhD, but it was actually during the Genomic Tools for Sweet Potato project where we really advanced in terms of developing, these tools for analyzing data in polyploid species. We are targeting sweet potato, but of course, it works for other species. This project was huge, involved multiple partners. Some of them already left the project because we kept getting these support from Bill and Melinda Gates Foundation, and we are about to end the RTB breeding project phase 1 now to get another started next year. So very exciting. So what's the matter about with sweet potato? The orange flashed ones we usually have here in Europe or in the US are not as common in Africa, in sub-Saharan Africa countries, and either in Latin America or China, which is the biggest producer, even though they're healthier, right? Because they have beta-carotene, which is a vitamin A precursor, on top of the other things, right? Carbohydrates, fiber, vitamins, and minerals. But what's the problem? Sweet potato, as well as the other polyploid crop species, have been delayed in terms of using genomic tools for a while. And this is because they are polyploids, right? Which adds a layer of complication. So here, just to show an example of what a wild relative sweet potato looks like under the microscope, which is a diploid, and what a hexaploid looks like, right? So we are having these number of chromosomes multiplied. Sweet potato and potatoes are not the only ones which have these type of genome. We also have fruit crops, ornamental crops, and forage crops. And another type of polyploid, they're called allopolyploids, and we are going to see the differences in a minute. There are all other types of polyploids, and we know them, right? Coffee, cotton, other forage crops are allopolyploids, and grains as well. So we understand that it's important to study and to understand them, right? The definition I like the most is as simple as we understand that these two complete sets of chromosomes we usually inherit from mom and dad have undergone genome duplication, right? And this duplication can happen in 2 different manners, usually, right? One of them involves crosses, right, among different species. That's what gives us allopolyploid crops. So they crossed 2 different species. They've undergone genome duplication. Now their gametes, right, look like this. They have more chromosomes to share. On the other hand, the autopolyploid ones, they didn't need another species. They just had gone through genome duplication. That's how their chromosomes look like now in their gametes, right? Of course, some of these characteristics of allo and autopolyploids arise from this phenomenon. One of them is preferential pairing, right? In the case of allopolyploids, they preferentially pair within the genome of origin, whereas in the case of autopolyploids, they can pair in other manners. We call them multivalent, right? So here just illustration of what an allopolyploid karyotype looks like. We can see the separation, right, among the A, B, and D genomes in wheat, for example. So if we fish or gish them, we are going to be able to distinguish these different type of genomes, right? Whereas in the case of autopolyploids, and this is an example of tetraploid potato, we cannot distinguish among the 4 homologous chromosomes that compose each one of the sets, right? And because of that, again, they are pairing within genomes in the case of allopolyploids, but they can pair among the homologous chromosomes in the case of autopolyploids. And these are the type of inheritances we study the most, right? Because it's what happens with humans, for example, where 2 homologous chromosome pair, which gives us bivalent formation. This is exactly what happens in allopolyploids, right? So chromosomes from genome A are going to pair with the homologous chromosome from of genome A, right? And so on. In the case of autopolyploids, we are going to have what we call polysomic inheritance, where more than 2 chromosomes can pair. They can pair in trios. They can form bivalence, just like in the disomic inheritance, but they can also pair in forming quadrivalence, or they can pair forming a trivalent and a univalent, right? In terms of gamete composition, that bivalents are more interesting, right? Because they're well placed in the metaphase during metaphase, and they can be pulled. And this is the way evolution makes sure that every gamete is getting the same amount of chromosomes, right? The other configurations might lead to some problems in terms of genome composition in the gametes.
Guilherme,
yes.
This picture, I've never done this, proper study of this topic. There is something what's called cytogenetics. Yeah.
Yeah.
So what is really a valent here? Can you point with the finger on?
So they're counting the types of pairings, right? So in this case, this duplicated chromosome, right, is paired with this duplicated chromosome, and once they pair, they can exchange pieces. That's what we call recombinations, right?
Yeah.
Whereas this one is paired with this as well as with this, and this one is paired. They're all paired, right, in different manners, and this one, they're only paired here and here. And this one is not paired with anything. So we are basically counting the number of chromosomes involved in the pairing, during pairing.
So the top right one is the univalent, and the bottom is the bottom.
Yes, exactly. This is the univalent, and this is the trivalent.
Okay.
Yes. Okay. So it's basically how they interact, right? I'm going to show the from our results one of these realizations, like when the hexavalent happened, for example, in the case of sweet potato. So that's where we start thinking about genetic analysis, right? Because we've been using SNPs for a long time, but the problem is that they are biallelic, right? So that reminds us of the big A, little a we used to study before. In diploids, we know what's the proportion of gametes there in big A and little a, right? And the proportion we expect when we self a heterozygous individual, right? Which is the 1 to 2 to 1 proportion. In the case of autotetraploid species, where pairing is happening randomly, right? We have this type of expectation here when we are crossing 2 double doses or selfing 1 double dose individual. And in the case of allotetraploid, we have a different type of segregation, right? 1 to 4 to 6 to 4 to 1. because they all depend on the proportion of the gametes that each individual can produce. So having mapping populations, right, individuals crossed is one way of detecting whether individuals are autotetraploids or allotetraploids or polyploids in general. Because we have these expectations in terms of those proportion, right, segregation proportion. So we had to come up with ways of obtaining what we call dosage, right? The same thing happens for diploids, right? We have 0, 1, and 2 representing the homozygotes individuals, right? 0 and 2, and one heterozygote individual. And if we think about it, they are diploids, so we expected 3 classes, right? In the case of tetraploids, we are going to expect 5. In the case of hexaploids, we're going to expect 7 genotypic classes, right? The problem is if we are using qualitative techniques like gel-based bands, right, we are not going to be able to distinguish, right, the dosage of each one of those bands, right? So let's say the 0 represents the absence, anything else is going to be presence, right? We used to have that problem in diploid as well when we are using dominant markers, for example. Whereas the dosage is going to allow us to count the number of reference alleles, right, respective to an alternative one. And we usually have these special names for these type of genotypes, right? Nulliplex, simplex, duplex, triplex, tetraplex. There are several ways of obtaining the information to be able to infer the dosage. One of them is using chip arrays, and then we use the proportion of signal to estimate the allele dosage. But we really like, because it's easy to obtain second generation sequence based reads, right? And what we do is, in fact, count the number of reads for each respective allele and infer the dosage from that amount of reads. Of course, we need to adapt the platforms, right? We need a higher depth to be able to estimate those quantities well. We've developed some tools and used them in several data sets. In this case, one of our partners generate modified GPS protocol they call GPS poly to increase this read depth. And then we use VCF with stored read counts, and then we run into some other functions or tools to be able to obtain these SNP dosage, right? So this is like easy to obtain now once we get the results, but such you have an idea, this is an example of a biparental population of Lipomia trifida, right? That wild sweet potato, diploid sweet potato species I mentioned. So if they are diploids, we have 3 possible types of genotypes, and this is what the results from GPS look like. So we are going to have either the allele fixed for little a or big A, or they're going to be heterozygotes, right? Easy. But then we have an example of potato data, right? So potato is a tetraploid species, so we have 5 possible classes, right? From 0 to 4 number of seeds in this case. And then if we look at the same type of plot, right, we are going to see the ability to distinguish the 2 homozygotes, again, either fixed for one allele or the other, and 3 different heterozygotes, right? Heterozygote classes. And in the case of sweet potato, we add some more alleles there. It goes from 0 to 6, which means that we are going to see something like this, right? Fixed for either allele or 5 different classes of heterozygotes, right? So as you can see, the nice plots we get are when we really increase the number of SNP, or the read depth, right? There is some noise here and there, which gives us some uncertainty in terms of which class the individual is from. But then we can also compute and use these individual probabilities in later applications. So just to give some examples, here are two mapping biparental populations we used in the development of those tools. We have the potato example and the sweet potato example. 154 individuals genotyped with the chip array, 8K SNPs, and then evaluated over 4 years for yield and quality traits. Same thing we did for the Borigat by Tanzania population that NC State generated, SID Peru generated. With 315 individuals, we used GBS poly, ended up with over 1 million SNPs, which were of course filtered, right, because of those read depth. And then yield and quality traits also evaluated over 6 environments. Just to give you an example of what a linkage map look like in a tetraploid species, looks like what we are used to see in diploid species as well. The recombination fraction shows those linked markers, right, with smaller recombination frequency, and between chromosomes with higher recombination frequency, right, close to 0.5. And then, so this is what we usually see analyzing diploid data as well. But well, we are not used to see something like this, right? Because building a linkage map involved is not only estimating the recombination fractions and knowing which markers are linked, but also knowing their phases, right? So this is basically what we are building here. For each one of those markers, which have like different doses, we are gonna have to place those variants into each one of the homologous chromosomes, right, or haplotypes. So this is the case of this population for one chromosome, let's say 1. But here is what is mostly interesting out of this type of analysis. We can visit every individual and know exactly where the recombination happened, right? And in the case of the first individual, for example, having a haplotype inherited by this individual with 3 different colors means that at least a trivalent happened during that pairing, right? And then we can visit every individual and see what was the contribution for chromosome 1, let's say, for from their parents, right? So A through D are representing the 4 homologous chromosomes from parent 1, and E through H are representing the 4 homologous chromosomes from parent 2. By looking at the results, we can also measure the level of multivalent formation. So we know that they vary according to the chromosome size, and then we can see that they can form quadrivalents, in fact, but in a lower proportion in comparison to bivalent formation, right? And this is, again, it's out of the observations we got, right? Because they might have had bivalents, but they could be quadrivalents. The problem was 2 of them didn't recombine, so we missed the opportunity to recognize quadrivalent. So this is at least this number of homologous chromosomes involved during pairing. There could be more. We can also measure out of the comparing with the probability of random pairing, if they're actually pairing randomly, all the possible pairs, or if there are any preferential pairing, right? Which is not the case, right? They are randomly pairing here. They're all close to what we expect for a tetraploid individual. We've analyzed phenotypic data in this paper, and based on these genotype probabilities of each one of the individuals, right? So here we have the 35 possible genotypes, right? When we cross 2 tetraploid individuals, each generates 6 different gametes, and when we combine them, we get 36 possible genotypes. And this is actually representing where the recombination happened, right? So it was AC from parent 1 and FH from parent 2, and then the A became D because of a recombination. So that's why most of the chromosome is actually this haplotype, and then it changes again out of another recombination, right? Or recombination that actually happened in the other parent. The same way we can get these probabilities for one single marker, let's say the first marker from chromosome 1, and see which ones are inheriting exactly the same genotypes. This is important because if there is a QTL specifically close to this marker, right, all these individuals are supposed to look the same, right, if the QTL is major, and if they inherited exactly the same homologs, they are supposed to look the same. And this is actually the strategy we used for QTL mapping, right? We can search for these regions of DNA that contributes to variation of a particular trait by looking at correlated QTL effects among individuals, right? Because we know that if they are shared by more than one individual, they've been shared through identical by descent, right? And remember, we went from the biallelic information, dosage information, to a completely informative setting, right? So now we know exactly which homologous or fragment of homologous chromosome was inherited by individuals. And the higher the identity by descent among individuals, the more similar they are going to look like, right? For one place where QTL is present. So what we did, we basically modeled the QTL effects as a random variable. We have a variance component associated with it, and then we test whether the variance component is different from 0 or not, right? We used a scoring statistics to be able to test quickly these variance components. An interesting fact is we've had other options of analyzing QTL before, but they are all relying on fixed effects for QTL, which means that building a multiple QTL model would take a lot of degrees of freedom to estimate them, right? But which is not the case when we're having one variance component per QTL. As an example, we have QTLs detected along these chromosomes for potato, and we have these big ones of chromosome 5. Some of them have been described before and some are new. We can basically use and assess those regions, try to develop markers and use them as or for marker assisted selection, right? Foliage maturity is, by the way, correlated with plant yield or tuber yield, and it's likely because of this common or shared location of these important QTL. So that was the example of the potato population. Here is the hexaploid sweet potato population. Again, we used our tools to build a linkage map. In this case, we ended up with over 30K SNPs. And this is what the linkage map for chromosome 8 looks like, right? We have these 6 homologs for one parent and the other 6 homologs for the other parents, right? So our work is placing these variants, right? Reference and alternate alleles in these specific locations in such a way that it makes sense given the population data, right? Which is represented here. So what we do is basically try all the possible options, right, for each combination of markers. And in order to do so, we use hidden Markov models in such a way we can have probabilities associated with it, right, and ultimately use it for analysis. So that's basically what we did. By looking at the inheritance pattern, we can see again a case of hexavalent formation for the Beauregard gamete. And here I think we had at least a trivalent formation, right? Again, it doesn't mean that the other ones were not involved. They just didn't recombine, or they didn't show any crossover, so we cannot assess that. So we've analyzed phenotypic data for the sweet potato population, again, we found these hotspots for several quality traits like dry matter, starch, beta-carotene, glucose content, as well as flesh and skin color. So we have these chromosome 3 and 12 with QTL for beta-carotene and flesh color and sugar co-localized. And these in another position, right? We have the control for skin color, which explains a lot of the phenotypic variance on a single QTL. These are all different, forget to mention the other slide, these are all different environments they were evaluated. We realized by looking at the annotated hypomia-TRIF, the wild relative genome, as well as RNA-seq data, that these are likely the case of linked genes. In the case of linkage group 3, again, as we saw, there are like QTLs associated in the same location for dry matter and beta-carotene, but they are negatively correlated, and this is likely the explanation, right? We need to break the linkage between these two genes in order to obtain something different of what we had in this population. And the linkage group 12, there's the gene called orange that works as a switch for chromoplastic biogenesis. It seems that if the carotenoid is produced but not stored in certain chromoplasts, they're going to degrade, and maybe the root's going to end up yellow but not orange. And here's an example of what's in fact happening for these two QTL on chromosome 3 and 12. At the same time as the dry matter decreases because the individual inherits a haplotype T from Beauregard, for example, it increases the level of beta-carotene. Same thing is happening for linkage group 12, right? Shows the decrease of some of the inheritance, right, of these specific haplotypes T and L for chromosome 12, shows that the value decreases for dry matter as it increases for beta-carotene. And that explains why it's happening, or the negative correlation is coming from. We validated these loci with for flesh color and skin color with a diverse population. It's actually a breeding population, genotyped only with 2.7K, and still were able to find, the same QTL on chromosome 3 for flesh color, but not the one for chromosome 12, and the same loci for skin color on chromosome 12, right? So with that in mind, we decided to come up with markers to be able to use them in marker-assisted selection, right? In such a way we can select in early stages without the need to wait to see the actual root and their color or other characteristics. At first, I used the skin color as an example just because it was easy to obtain the data for validation. But it turns out they actually liked it because the skin color is the only thing we see when we are buying sweet potatoes in the supermarket, right? So people decide whether they want that sweet potato or not based on the, skin color or some of the information they see. So here's just some examples of marker validation. I selected the highest R-square to show on that side, represented by A. Each one of them was tested for additive and dominant effects. And here below are represented the genes where SNPs were selected for validation, right? For validation, we used a completely different population from that one we used for discovering the QTL, right? We wanted to see if the QTL was still present in diverse populations or breeding populations. So this is an example. This is an example for flesh color. This is an example for skin color. So for skin color, you get a nice square of 0.49. Also seems to be additive. And yes.
In the plot subplot A, you have 3 progressions, if I see so correctly.
Yes.
There's one going from 0 to 6 with the dosage. What at the bottom?
Those are the additives, right? So we are basically adding one big A from 0 up to 6. The bottom ones are representing the dominant ones. As you can see, you have no zeros on the additive case, right, which is representing every class, so we cannot compare 0 against the rest of them, but we could do it for the other one. It still explains a lot of the variation, 0.35, but not as much as knowing the additive effect.
Just to be clear, the bottom two are for dominance.
For dominance, yes. One, it's 0 against the others, and the others is 6 against the others.
Did you ever look at? Yeah.
We wanted to know if the individual had at least one big A, if that would make a difference or not, or little a, right? You're going to ask something else?
So is this an example of, this is not an example of digenic versus trigenic dominance, right?
No, not really. So that's an interesting question. Well, the only thing we can actually measure out of biallelic SNPs is digenic, right? Because we only have 2 alleles. Going for other type of intra-locus interaction would need to know the haplotypes, right? And then pair them accordingly and test whether that has an effect or not.
So even with your allele dosages that go from 0 to 6,
yeah,
you cannot.
Because we are basically using the SNP alleles as a proxy for the QTL alleles, right? But we know that the QTL alleles might be multi-allelic, right? But we can all observe them as biallelic. And this is to make things simple, right? Because I know where you're getting. If we had more SNPs at that same location, and then we are able to figure out the haplotypes, then we could test other ways of genetic control, I guess. So this is the case of firmness, which is interesting because we actually design SNPs for beta amylase, which is an enzyme that gets active when you cook, and then it gets the root softer. So it's negatively correlated, right, with the firmness. But the correlation is only minus 0.4, which means that we could only explain about 20% of the variation out of one of the SNPs, and it was actually dominant, supposedly dominant effects that is acting here. And at last, we have trimeta with R-square of 0.24. Awesome. We kept doing this with other populations. This is the reciprocal, right, of this population we've been using. Tanzania is known to bear genes for nematode resistance, and of course, nematode attacking a root is not good for sweet potato, right, because that's what we eat. And it's really hard to get this type of phenotype, right? It needs lots of experiments. So we detect a QTL on chromosome 7. Yes, explaining 58% of the total variation. And it seems that you would have to have at least one of these haplotypes, H or I, to be able to be resistant to nematodes, right? So we did the same thing. We built SNPs for this population. So it's actually people from NC State and University of Georgia who developed those, and then we tested against their breeding populations, and then we saw that the dominant model was actually better as well. It gives us a R-square of 54, 0.54. Yeah. Now trying to implement all these tools in a regular manner in the breeding programs, either at NC State or in Africa or Peru. Our next step is allowing people to run genomic prediction models, right? We've tested it with the data we had at that time, which was the biparental population I was talking about. We were interested in knowing whether the QTL we found would be able to predict it better. And that's what we did here, comparing it with the GBlood Oh, and of course, we are like running the QTL search for every training population, right? Getting the QTLs found and then fitting them into the model in a model you call QBLUB or QTL BLUB. And found that for at least for quality related traits, which usually have those major QTLs I have shown, we can see that it actually increases our predictive ability, right? But when it comes to yield related traits, we do exactly the same search. The QTLs change, right? Every training population, and then we can see that it doesn't make much of a difference for yield related traits, which makes sense, right? Because they are usually polygenic. They interact more with the environment. They're not as heritable as the ones for quality traits. Another thing we were interested in looking at was whether if we use diploidized markers or the dosage markers we got, or the haplotype information we got out of the map, if it would make any difference in terms of genomic prediction. And in fact, we saw that haplotypes would give us a higher predictive ability for most quality-related traits. But again, it wouldn't make much of a difference in the yield-related traits, except for 1 and 2. But still, we understand that getting the dosage ready and done, it's better than having only the diploid-like markers. Some people have published that it doesn't actually make much of a difference. But I guess it has a lot to do with our ability to call in the dosage well. If the dosage is just called, but they are not very good in terms of quality, right? They might just perform the same as diploid-like markers. These actually, of course, we are now working with breeding populations and trying to see how we can apply these things. We can use diploid-like or dosage markers for breeding population, but what about haplotypes, right? And that's why I came here to the Roslin Institute, to the Highlander lab, to try to find a way of storing these type of information, right? Haplotypes in such a way it could be run efficiently inside a breeding program, right? Because these are only academic type of examples I gave, right? But when it comes to an actual breeding population, this population are much bigger, and they are going to go through several generations, right? And we need to have this information all together to be able to use it. So the idea here is to store this haplotype information into succinct tree sequences, which is a collection of tables that gives us information about accession recombination graphs, right? We are basically storing coalescence, branching, mutation, and recombination events within those tables, right? I'll give an example of what those tables means with our potato breeding population from SIP. Dave crossed several individuals, 4 males and 14 females. We selected around 1,000 individuals to get it started, and these individuals were all genotyped with this 2.5 SNP panel. We build the linkage groups integrated across all families, right? There are multi-parental families. We need to make the haplotypes talk to each other. Some of these parents are shared. We get the haplotypes probability, as I showed before, showing where the recombinations happened, and then we store these recombinations into tables, right? So if you're not used to tree sequences, we usually have to have these 3 tables: individual table, node table, and edge table. The only actual difference from what we've seen the examples they gave in using the TS kit is the need of 4 rows per individual, right? Each representing the 4 haplotypes. And then in the edge table, we are basically showing whether there was a recombination or not, right? So if it's chromosome 1 and it has 185 centimorgans, this one didn't have any recombination, but then these were recombined, right? Part of the homolog was inherited or was offered by parent 15 or the haplotype 15 of one of the parents, which is this one here, right? And the other piece of this haplotype was offered by the node 12 of parent 3. And then we do this not only for 3 individuals, which is our example here, but for all of them, right? Once we have the tables ready, we can represent those 3 sequences as nodes and edges, right? Representing this inheritance. A new tree means that there was a recombination, right? In order to get a new tree. And this is basically showing the directed relationship, right, among nodes or among haplotypes from parents to progeny. So we use this information to compare with the PCA we've got using dosage. We can see, which is in on the left side, right, or the plot A, and the plot B has the genomic relationship matrix from 3 sequences, right? So we can see that the locations, right, of these families are more or less the same, but with much less noise, right? Which is nice. And now we are interested in seeing how it performs in terms of prediction. Exciting. I haven't mentioned before, but this is how, just leading us to the final remarks, this is how the typical breeding programs for clonally propagated crop works. This is an example of potato, but it works for some others with some modifications here and there. So we cross these 15 to 30 parents, right? There are only 2 represented here, but there are actually many. There are multi-parental populations. We plant the 2 seeds, which are, each seed is a different individual, right? Just like animals. We plant them in nurseries. We selected them based on some criteria to plant around 2 to 2.5K individuals in observational trials. So during observational trials is when we are going to select for those highly heritable traits, which means that as soon as they have leaves, we can extract the DNA, run those markers for marker-assisted selection, and already eliminate some of them if they don't have the traits we want. And then we go to the preliminary, and then we start to evaluate yield, right? We plant these selected individuals in more than one location. We might observe them in more than one season. If you're in a tropical country, you can actually plant sweet potato twice, for example, in a year. And then we have observations about yields. The propagation material is increasing, right, as we are getting more and more roots. And then we keep evaluating them, increasing the number of seasons, the number of locations. We are observing those up to the point where we release the clone. The genomic, I mentioned that where the marker-assisted selection would go, right, it's easy for us to genotype 2K individuals and get information about those markers I mentioned. And maybe more sustainably, we are going to be able to run the genomic prediction for the preliminary yield trials as we are still increasing the database for prediction. Running 2.5K SNPs in 500 clones is also easy. But what we are doing now for the 3 sequence work is genotyping the 2K individuals, right? So our whole seed populations are bigger. They allow us to get the haplotype information, and then we want to see how it performs as these cycle of parents run year after year, right? Which is pretty exciting and interesting because it gives us this opportunity, right, to study this inheritance across a closed setting. So just as lesson learned, we have this idea that marker informativeness matter. So we understand now that haplotypes bear more information in comparison to dosage, and they have implications in terms of, QTL mapping and genomic prediction. The turnaround time also matters. So we worked a lot to be able to get these platforms working and running. Because again, the decisions have to be made quickly by the breeders. Get to know your traits and create a strategy on how to make it. This is our idea now. We target oligogenic traits by means of selection, of marker-assisted selection in early stage. For example, we're looking at beta-carotene content on nematode resistance, and use genomic-enabled prediction for these highly polygenic traits in later stages, right? Such as root yield and texture. Now, how will tree sequence perform in the future? This is our question. We need to get MapPoly version 2 working for multi-parental cross-generation mapping tree integration. This is something I'm gonna be working towards next week when I go to NC State. And we also want to see, using AlphaSigma, how prediction accuracy and marker density is affected by the information of haplotypes, right? As well as whether it makes any difference or not, because, well, it's an interesting question, isn't it? We go through a lot of additional work to be able to obtain the haplotype, so it needs to matter in terms of genetic gains. So this is something we are looking forward to working together soon. I would like to thank the people who offered their populations and data sets for us to work with. NACRI, SAPE, and NCSU especially, the analytical support from University of Edinburgh, especially Hannes and Gregor, and for having me here during the sabbatical. And people from CIMIC and NCSU have also been helping us to analyze this data, and my current team at UFV. Some of them I don't even know in person yet because I was here when they got to my lab. But yeah, we've been talking remotely since. Thank you.
Thanks, Guy. So to start off with our, if anyone has any questions, for the people online, if you have questions, raise your hand, turn on your microphone, or just type in the chat. And is there anyone in the room who has questions?
Very quick one. Yeah. So you showed the results of the QTL-based genomic prediction models and the dosage-based genomic prediction models. And simple question: have you thought about just combining the two so they've got both kernels in them and just estimating variances?
We did. I just dropped them from the results because it didn't make a difference for both sets of traits we had. The quality-related traits, they're always good, either with QTL or QTL plus the G matrix, right, for the rest of the genome.
Yeah.
There wasn't any increase in terms of predictability. And the same thing happened for the yield related data. It didn't increase when we added the G matrix. It was basically working kind of the same way.
So it performed like the QTL one in both
Right.
scenarios.
Yeah.
So it didn't leverage the
best. It didn't. And again, this is a limited scenario, right? Because we are looking at a biparental population, which is not reality for breeding programs, right? Or not only one of them, it's actually a bunch. But in order to get the QTL out of haplotypes, we would need to have the map built and when the QTL detected, and of course we can do it by means of GWAS, right? We can run GWAS and try to see if we can get those peaks, but having QTL found for yield-related traits, it's going to be really hard to obtain. So we just, for now, we are not targeting that, combination yet. Maybe it's going to be important in the future once we have, them all haplotyped, linkage maps built, all phased into tree sequences, then maybe we're going to see something there. But yeah, long way before we see it.
So I was really interested to see the sample sizes that you were using and working with, and maybe you can explain for polyploids what are the ideal numbers for sample sizes? Because in the linkage mapping, for example, it was one cross, but hundreds of full SIBs.
Right.
And then for like the marker assisted selection, it was like 244.
Right.
So I feel from like my perspective, those are fairly small numbers.
Right.
Maybe you can.
Yeah, for biparental population, those numbers are actually okay for detecting QTL. Maybe the effects are biased, right? And we know that it's going to happen anyways because of sampling.
Is it because you're really looking just at like one specific line or?
Right, yeah, so for building a mapping populations, we need to cross these divergent individuals, right? One is orange, the other is white. They're going to be segregation in the population, then try to find the region that's controlling that. So for major QTL, these numbers are okay.
Mhm.
For building the multi-parental population integrated maps, right? We are targeting populations up to 50 individuals per full SIB. And during simulations, we can do even less, right? But again, there is not the noise we expect in real data. So 50 seemed okay for tetraploids, right? We are doing it for hexaploid or the hexaploid multiparental populations. And there we need more individuals per full SIB to be able to run these. But it's also interesting because if we can barely get a map, once we put all the families together, they're going to help each other in terms of, phasing and market distance estimation. So it gets interesting, like when you put them all together as well. But yeah, we'll see. Thank you.
Guy, when you showed your crossing plan, some crosses didn't succeed, or you targeted in this way?
Well, this is one thing about autopolyploid species. Some of them have like very strong problems with fertility. It seems to be related with self-incompatible genes. It's very common. And it makes sense, right? Because once they undergo a selfing, there is a strong inbreeding depression. And then to avoid that in nature, I guess, they have these bunch of multi-allelic series of self-incompatible. And then you don't know in advance, right? Once you are trying the biparental crosses for the multiparental population, that's why some of them fail completely. They might be incompatible, and therefore no seeds are generated. So we have these highly unbalanced, right, representation of parents. We understand that it could be a problem and biased genomic prediction, right? Because some of them are going to be more evaluated than the others. But again, maybe tree sequences is going to help with that too, right? Because if we are able to get back to the past, right, and understand where they're all coming from, this could be more information for prediction. But yeah, to be seen.
Could you use, so I guess you're not seeing progeny that are probably homozygous at certain positions.
There could be homozygous in certain positions, yes.
Given the the genotypes of the parents,
yeah,
you know roughly what proportion of individuals progeny you expect. Homozygous, heterozygous, all the different genotype classes.
Right.
You can work backwards to which loci are showing this, this incompatibility.
It really depends on what the parents are, right? I like the idea. Maybe we just don't have enough representation from parents and progenies to be able to detect that. It's basically looking for runs of homozygosity, right? Looking for these lack of homozygosity. No, yeah, I think that's a good idea. Like to try to find right where the incompatible loci are.
This was a very useful strategy in animal breeding to find lethal haplotypes. So these are sort of embryos you never see.
Right.
Because they usually die,
right.
in uterus.
Yeah.
Yes.
I like it. Yeah. Oh, yeah. Let's talk about it. I think we have enough data to try to find it now.
Any final questions, comments? If not, then thanks again, Keith.
Thank you.